Some scientific stories begin with a breakthrough, but this one begins with a disaster. In the late 50s, thalidomide was a drug prescribed to pregnant women to cure morning sickness, but it caused several birth defects, causing one of the biggest issues in the history of medicine. Decades later, scientists discovered something remarkable about thalidomide. By studying how this molecule works, they discovered a new way of tackling other diseases. Instead of simply blocking a harmful protein, they could mark it and use their own cell machinery to destroy it. A recent review by two research groups at IRB Barcelona brings together all this family of drugs. I interviewed the main authors to know the story and the future of protein target degradation. So, 30 years ago, how did we develop drugs, and what did the drugs do to eliminate proteins that cause diseases?
So for many years, medicinal chemists have developed small molecules that will bind very strongly to a target protein, a protein that has been identified to be bad for our own health. However, we have been developing new methodologies that kind of go far away from just blocking the target, and instead what we are doing is inducing the degradation of this target using the endogenous mechanisms inside of the cell.
So, if I understand correctly, it's our own body that destroys the protein that is causing the disease. Is that true?
Exactly. What we're doing is taking advantage of a process that happens naturally inside of the cell, and what we are doing is repurposing for our own benefit. We are able to know which proteins need to be degraded, so we just help the cell identify them.
Okay, how do you help the cell identify which proteins have to be destroyed?
So to do so, what we do is we prepare small molecules that are able to bind strongly to the protein that is in charge of marking which targets have to be degraded inside of the cell, and we change the selectivity of its selectivity.
So the cell then does the job and destroys the protein.
Exactly, we just have to tell them where to aim at.
What kind of drugs or chemicals scientists use to target these proteins related to diseases?
In the beginning of the field, scientists used peptides and small proteins, but these are not good drugs. So later, they found that thalidomide, that's been a drug for a while, worked as to hijack this cell machinery. And so we repurposed the use of thalidomide that initially was used to treat pregnant women for morning sickness, and then they found out that they were causing birth defects and fetus, and that was a tragedy. But now we could repurpose and then find new small molecules based on this thalidomide that are used in cancer treatment and other disease treatments.
So then this molecule thalidomide is now used as a drug, but for a different purpose, to target these proteins, and to which kind of diseases is useful.
Okay. So the thing is, you can treat any disease that you like, because this thalidomide binds to the cell machinery, but the other part of the molecule binds to the protein that you want to destroy. So you can target proteins related to cancer and to many other diseases. Just have to choose the protein wisely and know the mechanisms that in diseases, so you know that the protein you need to target. But you can use it for many different diseases.
And how many drugs based on this type are in the literature?
A really breakup, like recently the first protein target degrader has been approved by Arvinas, and it was quite big news. So they've been developing this for a while, and we were waiting for it for a while, and now it's in the market. Well, it's supposed to be in the market very soon.
So for how many years have been these drugs in research, not in the market, I mean in research?
The first one was 2001, more or less, the very beginning of more kind of proof of concept studies that the concept of degradation to treat diseases worked. And after that, there have been lots of developments, and now there's thalidomide, but they found really many other, well, not that many, but other molecules to use in this. I don't know, 25 years already, but it's quite a young, but not that young field now.
So you did this extensive review. What was the goal of the review itself?
So basically, this field over the last 2 decades has just exploded because of the potential of treating cancer. And so there's been a huge amount of research being done, huge amount of publications. So as a result, we need to compile this, generate a nice review, specifically on cereblon, which is the protein that we're talking about. So that's the part that can actually degrade our protein of interest. In our case, our lab focuses on degrading cancer-related proteins. So in this review, we collected just over 100 different ligands that bind to cereblon. And basically by providing this to the community, they can select specifically to fit their needs for what kind of PROTAC they want to develop. And in addition to that, they also, we provided some structural information. So there's actual images of these binders bound to the protein cereblon using X-ray crystal structures. And so using these X-ray crystal structures, we can actually detail specific interactions. And again, this is kind of helping researchers to tailor make their PROTACs. So specifically what they need.
So if I was a scientist that I wanted to develop a drug, this review would be very helpful for me. In which sense?
So it really helps you cut down on a lot of research yourself before you actually step into the lab and start doing synthesis. So again, it kind of just guides you and suggests things that you should maybe you missed when you were doing your literature review yourself. So it's just basically by categorizing it, you can kind of decide based on that.
Okay, thank you. Thanks for watching. If you enjoyed the video, follow us and subscribe to our channel for more stories.